polyacrylamide gel electrophoresis. I think we did discuss this in I think 311 course.
Yes, sir.
Right. Yeah. So there are two gels. One is basically stacking gel, and then below it is resolving gel. This is called vertical electrophoresis. Right. So you load this sample in stacking gel, and then you pass high current, right, only for a brief period of time, so that all these proteins that are present in your samples get stacked at the interface of stacking and resolving gel, and then you basically reduce the current, and then it is resolved slowly, depending on the molecular weight that the polypeptide chains have, right? So, and that is what is shown here in the next slide. Okay, so this is actually a stained gel from SDS page, and this dye they used was actually the Scumacy Blue Brilliant Green 250. Okay, so basically this can stain protein in a sample. When you have whole immunoglobulin G, we are talking about immunoglobulin G here, right? So you get a molecular weight marker of approximately, so this is the marker, molecular weight marker you are running here in this rail, right? And you already know that what are the different proteins you are including in your marker. So you have, let's say, 17 kilodalton, 26 kilodalton, 32, 48, 75, 109, and so on and so forth, right? Now, if you have the whole antibody molecule resolved in native form means without reducing it in any way, so you get 150 kilodalton range. So this is close, this is above 109, so this is actually 150 kilodalton. So in that range you get the molecular weight, right? Yeah, you get these small rather thin bands in this range. So this is because you there is always some contaminant in the whole preparation which actually is reduced. Okay, so now if you have actually reduced this antibody molecule immunoglobulin, right, so almost everything is reduced here, and now what you are left with is the heavy chain and light chain, right? Heavy chain is coming close to 55 kilodalton, right? You are basically reducing it. So let's go back. So this is what you are doing. Okay, you have the antibody molecule, you are reducing it. So you're going to get 2 chains of heavy chain and 2 chains of light chains, right? Because the size of each of those heavy and light chains actually is same. So this is 22, this is 22, this is 50 to 55, this is 50 to 55, right? So they are going to give you one band in the SDS page, because this is resolved based on the size. And if the size is same, so they will actually migrate at the same place. So what you got? You got one band corresponding to heavy chain, which is close to 55 kilodalton, and one band corresponding to light chain, which is coming close to this 25 kilodalton, right? Now, if you are very basically thorough, you are making sure that no other contaminants are present in your preparation. So then you can actually get these clear bands. Otherwise, so you have to look at the most predominant band. Okay, so you have 2 of those here, right? Now you have digested it with papain. So this is actually a papain digested slide. So what are you going to get? So you have digested this antibody molecule with papain. Now let's go back. This is what you did. So it has digested above this disulfide bond in the heavy chain. You are going to get one band corresponding to around 45 kilodalton, right? and 155 kilodalton in non-reducing condition, right? So so this is one FC portion, okay? And then you get this, yeah, yeah. Basically, this is reduced FAB region. Apparently, this was actually also added with this mercaptan dye, which actually can break these bonds. So you get two bands, one F for FC, one for, and FC actually consists of only the heavy chain portion, right? And this is the FAB, which also has part of the heavy and part of the light chain. How do you determine? Actually, this also has reactivity against the only heavy chain, and this only has reactivity against both. So the same trick, you generate this antibody against FC and FAB, and then basically do a Western blotting, right? So how do you do Western blotting? So basically you resolve this polypeptide, you transfer onto a membrane, and then basically you do all these steps, blocking and so on and so forth, and then you come up with these antibodies and light up with some enzyme substrate combination, right? So you can actually clearly see that this FAB, rather anti-heavy and light chain sera is going to light up these, but only heavy chain sera is going to light up this FC portion, right? Now, the other thing that you do is, so you have digested your protein molecule. Protein means the IgG molecule. Then you have actually passed it through this matrix, which is coated with protein A. Okay. So what is protein A? Protein A actually is a bacterial product. Okay. And this has affinity towards the FC portion of antibody. Okay, so it actually, so one of the ways how you can purify your antibodies is by running it through the immobilized protein A. Okay, you have this column, you have these beads, right? On these beads, you have immobilized this protein A or protein G matrix. So now if you pass your antibody, so because FC portion has affinity towards it, right? So antibody will remain bound. Rest of the things which are present in serum or other or the sample that you have, they will actually flow through, and then you can elute it. So this is one way how you can purify your antibody through affinity purification. It's not that always you have to use these nickel NTA, GST tag columns and all those things, right? So you can actually, at least for antibodies, you can use protein A or protein G columns for purifying. What was done here was actually you have passed this digested fraction through protein A column, and then you are collecting whatever is coming as flow through. So you can see that FC portion is not coming in the flow through, but the rest of it, this portion actually is coming, right? So this is actually FAB, which has the both heavy and light chains, right? So that actually is coming in the flow through. Now, then you wash this column multiple times, not much is left, and then you elute it. Okay, whatever was bound onto this column, so then you elute it using a reduced pH buffer, right? And then you can clearly see that with this process, you can purify only the FC portion of your antibody molecule, right? And this is actually for the non-reduced FAB. This is for the reduced FAB. This is not for the non-reduced FAB. All these steps were done. Flow through, and then the whole IgG molecule, you already know that this is basically coming here. And then you can actually also purify FAB as a control, the purified FAB if you load. So with this purification, you can actually achieve FAB and FC separately, heavy and light chain separately, right? Now, there are different matrix even to purify light chains. We are not going to talk about those, but this is the kind of analysis that was done to determine the structure of antibody molecule. Okay, and to demonstrate the functionality that which part of antibody molecule is responsible for binding to the antigen, and whether that part consists of both heavy and light chain, or it has only the heavy chain. Okay, so all those things was done or were done using this approach. Question on this slide? All right. Yeah. So again, so if you want to work on antibodies, and if because this was around the time and no monoclonal antibodies were available, right? So you have to actually have you either have to work with heterogeneous population, right? Population means basically you collect serum, and then you try to purify these antibodies from serum. So you don't know what it is reactive to, right? So all those things are there. There's some question. Can you explain the flow through lanes again? Which flow through lanes again? This one? So flow through means basically you have the digested material, right? You have actually passed through this, and if on the column, whatever the matrix you are putting in the column, so that was actually mobilized with protein A. You pass this digested material, right? Or basically the whole IgG, and when you do it for the whole IgG, actually this is how it was purified, just by passing it through this protein A column. This is a purified fraction, more or less, right? You digested it with papain, so which is going to cleave it above this disulfide bond in the heavy chain. So you are going to generate FABs, right? If you do not add mercaptoethanol, so you are going to get a molecular weight polypeptide band of 45 kilodalton, because both are same, right? But if you digested it, you are going to get around 20 kilodalton weight for this region. And for this region, the rest of the heavy chain will be you are going to get. Okay, so basically 50 kilodalton for the whole FC region, unreduced condition, and if you digest, if you basically reduced it, so 25, right? Now this is what you are getting here. With protein A flow through, in flow through you don't get the FC portion, a band corresponding to the FC region. Okay, so you get only the FAB, and that basically tells you that the FAB portion does not bind to protein A, because this actually flow through the column. And whatever was bound, if you elute it, so you will get only the FC, the purified FC portion of your antibody, right? And if you use it in non-reducing conditions, so basically the molecular weights are going to shift. Because with non-reducing, so you actually have two chains, and both of these chains will give you a higher molecular weight. Okay, close to 45 kilodalton, right? Where you can actually purify this FAB fraction from the flow through, because this was coming in the flow through, right? Is it clear or not? Yeah. Okay, so this is what I was saying. So usually, so many of these studies were done using a heterogeneous population of these antibody molecules, or heterogeneous species. Okay, so you don't have actually a monoclonal antibody reactive to a known antigen of your choice. Okay, so one good thing around that time these people they did was they somehow identified someone having a multiple myeloma. So this is actually a cancer of antibody producing cells. Although if somebody is suffering from a cancer, this cannot be a good thing, right? But for doing investigation, so they actually use sample from those patients. Now, if you have a cancer of antibody producing cells, so that means you will have one particular cell type which is going to give rise to multiple clones because of uncontrolled growth. And whatever the product it's going to secrete, so they will have one and the same specificity, right? So this was the sample which was actually used for purifying these antibodies of monospecificity. Okay, they were initially called as myeloma proteins, and if you actually collect this myeloma protein, so 95% of the serum in these patients actually will have only immunoglobulin, right? So this actually provided a good source of antibodies of unique specificity, of a monospecificity, that was used for this whole analysis. Okay, not initially, as I said, it was basically you have an antibody molecule, and all so many of these antibody, billion or trillion of those antibody molecules, but the specificity of each of those, you have no idea what they were reactive to, right? But in this case, so they, whatever the antigen they were reactive to, that was one. Okay, so they had monospecificity because they were coming from one single clone of your B cells, right? Now, for light chain analysis, actually, this is again a cancerous condition where this excess of light chains was excreted out in urine. These are called Benz-Jones proteins. Okay, Benz-Jones proteins. This was the sample used for characterizing light chain in the antibody molecule. Okay, experimentally, you can also basically generate these cancers using this mineral oil. Okay, so actually you can inject mineral oil in the peritoneal cavity of these animals, these mice, and after some time, so they are going to develop this cancer that we call as plasmacytoma. This is mineral oil induced plasmacytoma, MOPC, MOPEC, right? So this was from humans, right? But many a time, so you are limited with the amount of sample you can get. But with this technique, so basically you can actually inject mineral oil in animals, and then you can keep collecting the samples and characterize these antibodies to define what is the composition, what is the charge present on these molecules, what is basically which portion is able to bind to antigen, which portion is able to is responsible for carrying out the effector function of the antibody molecule, right? So all these analysis was done with the samples, which is actually highlighted on this slide. Okay, so briefly, we did talk about immunoglobulin domain or immunoglobulin fold, right? Where, when I said you have something like as this, right? This is called one immunoglobulin domain. Or in other words, this portion actually is also called as immunoglobulin fold. A stretch of amino acids, about 100 of those, 100, 110 of those, separated by this interchain disulfide bond. This is called immunoglobulin fold. In other words, immunoglobulin domain. Now I'll get back to the same slide. Okay, now immunoglobulin domain is what? So each contains about 110 amino acid residues and an intrachain, because this is within the same chain, disulfide bond that forms a loop of about 60 amino acids. So that looped out portion actually is 60 amino acid, and then you have some part here, and this is basically the disulfide bond. So this is called immunoglobulin domain, right? Now, these domains actually um, have variable region, right? As well as a basic when they are actually coming from this VH, right? Of light chain or of heavy chains. Okay. Now in this variable region, so you have a certain region. Again, I'll get back to the same slide as I referred to as earlier. So something like this. So these are some fixed regions, fixed region in this sense. So 3 in light chain and 3 in heavy chain, which actually exhibit high variability from clone to clone. And this actually constitute about 15 to 20% of the variable domain. Right? Now in this region, so what you normally have is these 3 regions that we call as complementarity determining regions, CDR. Okay, CDR1, CDR2, and CDR3 starting from N terminus to the C terminus. Okay. And these CDRs, okay. So let's say you're starting from N terminus here. So then you have one CDR and then basically another region and then another CDR and then third CDR. In between, so you have these framework regions. This is one framework, FR1, FR2. In between FR1 and FR2, you have CDR1. Then FR2 and FR3, right? Between these two, you have CDR2. This is how these are spread out in the variable domain of the antibody molecule, right? So these are regions where they do not show variability in the amino acid sequence from one molecule of antibody to the other molecule of antibody coming from different B cells. Okay, so these are called framework regions, and where you find a lot of variability. So these are called CDRs or hypervariable regions. Diversity in the VH domain is actually concentrated in the CDR regions. Diversity means what? The ability of these antibodies to recognize more than one antigen or unheard number of antigens if you talk about the complete repertoire you have. That diversity is provided by the variable regions. Because if you have the same thing in the framework region, so that is not going to contribute heavily in binding to the antigen. The portion which is variable will provide the antibody molecule the ability to bind to different antigens. Okay, so that actually is present in this VH domain. All right, this is actually what is shown. So for example, let's say if you sequence these several thousands or several hundreds or maybe even more than that of these antibody molecules, right? And you actually look at the extent of variability within the VH domain or VL domain. Now, when I say these terms, so are you able to understand what I'm saying? VH and VL, I think this should be very clear by this time, right?
Yes, sir.
Yeah. So what you are looking at actually is the position of these residues in the domain, in the variable domain, starting from 0 to 120. And same case here, starting from 0 to 110 or something in light chain, right? Now from and on y-axis, you are looking at the extent of variability. For example, at position 1, in different clones, which amino acid is utilized? At position 2, which amino acid comes up? What is the frequency of utilization of that one particular amino acid, right? So you can clearly see that there are only 3 regions in the VH domain and 3 regions in the this VL domain, which actually show a lot of variability in the usage of those amino acids. And these actually are called hypervariable region or HVR, right? hypervariable region, HVR. In other words, CDR1, CDR2, and CDR3. Now, these 3 CDRs coming from basically the light chain and 3 CDR coming from the heavy chain, they actually constitute what we call as the paratope, the site of the antibody molecule which interact with your antigen. Because there are a lot of variation in these. Now, if you have, for example, one clone, so it will have certain kind of these amino acids in at that fixed position, right? Now, in another antibody molecule, you have other amino acid utilization. And that actually changes the specificity of one antibody molecule from clone to clone and from one B cell to the other B cell. How do you actually determine this extent of variability? So this is the actually formula you use. Variability is equal to number of different amino acids at a given position divided by the frequency of the most common amino acid at a given position. For example, alanine is used here, right, as a very common thing, right? Now, out of those 100 or 1 million or 1,000, how many times actually you get alanine? And reverse of that. So here, for example, in one clone, so which you use as standard, so it was coming as phenylalanine. But in the other clone, so you find alanine, tryptophan, glycine, so on and so forth. So this is how you, and then basically you plot it that if you actually sequence multiple number of these clones, so actually you find that in this region, so you have a lot of variability, and in these regions, so you don't have too much variability, right? And these regions we know are responsible for interacting predominantly with the antigen, and this is what decides the specificity of an antibody molecule.
So,
yeah.
So can we say something if we compare the two graphs like of pH and VL, like there is a difference also. We see a crack also.
Oh, obviously. Rather, the most variability actually is provided by the heavy chain, limited by the light chain.
So what does the crack denote in the near 120? The crack, there's a in VL domain, we have a...
Yeah, but well, that actually depend on, for example, so you actually are looking at as a whole. Maybe this amino acid at particular position is responsible heavily for interacting. Okay, these are not that involved that much. So, and these numbering are nothing but Cabot numbering, right? Now, based on that, you decide whether this is going to actually interact more efficiently with your antigen or not, or rather, in molecular term, you call it as epitope, right?
So I was asking about the small 3 cracks we see around 120 units of variability.
Yeah.
So.
Yeah, well, this is actually only present in the VL domain, right? Where basically this it's extending way beyond 120 at one position. But why do you care too much about that?
Fine, sir.
If you actually overall, if you compare the average, so average variability actually is much higher in CDR1, CDR2, and CDR3. Okay, and here it's going actually up to maximum up to 50, and then largely on an average up to 30 or 25. And now, for example, this is one of those examples. If you sequence millions of those clones, probably this will even get normalized even a much better way. Right?
Fine,
but for sure, it's not going to give, maybe this will give you actually a very basal line. Okay, but still you get some variability here, very clear variability, and then you have nothing here, almost a flat line, and then you have variability in this region, and then flat line. That actually depend on the number of sequences you are analyzing. Yes?
Fine, sir. Yeah, got it.
Okay, again, domain interaction. So this is actually what we are talking about here. Now, um, if you actually look at this slide, so this shows which domain interact with the other domain, right? So, for example, VH domain and VL domain interact. Okay, CL domain and um, the CH domain interact, CH1 domain interact. Okay, there is one domain which is actually clearly shown here, that CH2 domain does not directly interact with the CH2 domain, and the simple reason is because this has those amino acids which can be glycosylated more efficiently. So, so all those amino acids which can be glycosylated are present in CH2 domain, this carbohydrate region. So basically, this carbohydrate, a lot of these glycan motifs, these carbohydrates actually come in between these two domains. So they don't actually, amino acid side chains do not interact, right? If you actually make a crystal structure, so this is how it looks like. This is not like a single chain. Basically, you have actually drawn a line or something like that, right? So these are actually different domains, right? VL, CL, VH, CH1, CH2, CH3. So this is basically to show you how this molecule looks like, and where actually the binding with the antigen actually is taking place only at this site, at the tip of this, at the very tip of this VH and VL domain. And the area which actually interact with your antigen or epitope are the one which are projected, and they actually are constituted by these 3 CDRs from the VH domain and 3 CDRs from the VL domain. So in one antibody molecule, so you'll have 2 antigen binding sites, right? If it is a basically IgG molecule. For example, it's a pentameric IgM. So again, you will have 10 of those. So probably it will bind much better in terms of its avidity, right? But affinity is always better for IgG antibody, because this is formed after class switching. So again, I'm introducing these terms, but I'll talk about those things in little more detail. For the time being, remember, CH2 domain of one heavy chain and the other heavy chain do not interact. Every other domain actually interact. And at the tip of this Y-shaped structure, you have the antigen binding site. 2 of these are present in a conventional normal antibody. Okay, so this is basically how this antibody, the FAV region, of this antibody, one is giving rise to this light and coming from the light chain, and one is coming from the heavy chain. And this is basically, well, one of them actually is coming from light chain, one is coming from the heavy chain, and this interacts with the antigen. And you can actually clearly see that there is a complementarity. For example, where you have a dip here, so the antigen is going to be projected. Where you have dip in your antigen, so here the paratope is going to be projected. So you kind of establish more or less a complementarity between the interacting surface of antigen and antibody molecules, right? Yeah, so this slide actually show you nothing but the flexibility, or basically the adjustment that an antibody does after binding to the antigen. Okay, so before binding and after binding to the antigen, so you actually can see that there are some changes. It's not like if you have a native antibody molecule, or purified or separated antibody molecule, isolated antibody molecule, so it will have exactly the same configuration. Okay, the configuration will be different for the antibody molecule which is alone or which is bound with the antigen. So this flexibility is there, and this is basically to provide a snug fitting, okay, so that it can actually bind efficiently to the antigen. So this is what is shown here. CDRs coming from light chain L1, L2, L3, and from heavy chain H1, H2, S3, right? So there is some adjustment when an antibody molecule bind to the antigen. All right, so we already know by this time that you have this antigen. On this antigen, you have different epitopes. This epitope can be bound to one antibody molecule, and the other epitope on this antigen can be bound by another antibody molecule. Okay. In molecular term, you actually talk about the epitope and paratope. Okay, not antigen and antibody interaction, because within an antigen-antibody interaction, you have multiple epitopes and paratopes, right? So this has one different specificity. This has a different specificity. This antibody is going to bind to this particular epitope, right? And this is going to bind to a different epitope on the antigen. One more thing that you have to understand, or actually I did refer to it, that when this antibody molecule is considered as BCR, which is actually present on this membrane. So this has modification in the, this actually has a complete C-terminus region, right? But once this is secreted, some part of it is cleaved, particularly the portion which actually is traversing through the membrane, and the one that has the cytoplasmic domain or cytoplasmic tail. So that actually is chopped off, and this is actually achieved by this proteolytic processing once the antibody is formed after translation, right? And then this antibody is secreted by these B cells, and they can actually then be found in extracellular spaces like blood or other tissue fluids. So change is not at the N-terminus. Okay, change actually happens predominantly at the carboxy termini, because the specificity remains same. Okay, whatever the specificity of BCR was there, so this remains same for the secreted product as well. Okay, but the C-terminus is changed. Okay, what is this actually? Although it may not be directly interesting to you, but many a time actually, when you use an antibody for your biological analysis, right? So you actually have to characterize, or you have to obtain a clone of antibody which is characterized to be either blocking or signaling antibody. Okay, let's say I'll give you one example. You have an antibody against a surface molecule, right? You have a cell surface, you have a molecule which is kind of exposed here, and you use an antibody against this. Now, you treated, or you basically you are having your cells, you added your antibody to these. Now, there can be two things that can happen actually. One is that this antibody somehow binds to the site and it blocks the interaction of this antigen or this receptor with its ligand. Okay, so this would be called as a blocking antibody. And signaling antibody is, for example, you have this receptor, and this is the site where actually if ligand is bound, so this will trigger signal. Now, if you chose an antibody which also bind exactly at the same site, it could be that it will serve as the ligand, and it will trigger signaling within the cells expressing that receptor. And this can be actually shown in different ways here. Okay, so one is, now we are trying to distinguish between blocking antibody and signaling antibody. Okay, blocking or antibody is going to be the one of those, rather, A to D, and signaling is basically shown here. How? Let's say you use this antibody, and this bind actually to the ligand. Now ligand will not be available to binding to this receptor. This is receptor in the membrane, right? You have these different domains, and in domain 3, for example, you have the site for binding to the ligand. One thing is that ligand is sequestered by this antibody, and if this ligand is sequestered, so you actually can block signaling. Second thing that can happen is somehow this antibody, so you can select an antibody against the receptor which actually bind outside the pocket, and it somehow restrict this site from binding to the antigen. Both of these antibodies are going to serve as blocking antibody. Other type of antibody which can actually act as blocking antibody is, let's say you have two chains of a receptor, and in order to induce signaling, so they have to come close, and then you have this interaction between these two chains, and then you transduce signaling, right? Now if you use an antibody against a region which is responsible for this interaction, okay, this domain, for example, this chain of the receptor and this chain of the receptor are responsible for interacting each other, and eventually so they transduce signaling. And somehow if you sequester these sites on the receptor, so again these antibodies are going to serve as blocking antibodies. Another thing is basically this antibody, if bound to the receptor, can change its configuration, and because the configuration is changed, now it cannot really interact with this ligand, or alternatively it can make it more functional, right? But let's say if it is actually making the site unavailable for interacting to the other receptor unit, so this will again serve as the blocking antibody. And the other example I gave you was a signaling antibody. So this actually will serve as the mimic of your ligand, because this is also going to bind to exactly the same site where your ligand is binding. Now, why is it important to know what is a blocking antibody and what is a signaling antibody? So you might have heard about, or let me frame it differently. So who got Nobel Prize in 2018? Probably you don't remember. In immunology, rather in physiology and medicine, so these two guys, Tasuku Honjo, and there was another guy from Texas. So what was his name? Jim Ellison. So they got Nobel Prize to show immune checkpoint therapy. Okay, particularly for managing different kinds of cancer. Okay, so what they showed was they actually used these antibodies for binding to certain molecules on the surface of your lymphocytes, which are specific to one antigen or the other. And if you do that, somehow the inhibitory interaction of the receptor expressed by these cells with its ligand is blocked, and in so doing, so these cells now become more functional. Okay, so in that case, actually they use blocking antibodies. If, for example, if they could have used a signaling antibody against that inhibitory receptor, so phenomena is going to be different. So you actually induce more negative signal to your cells, and in so doing, you would actually harm these cells more. And even for any analysis that you perform in your laboratory using antibodies, when it actually relates to the functionality determination. If you want to find out whether this is an antibody against a particular receptor, and we used it to block this signaling through this receptor, you actually have to make sure that the antibody you are using is a blocking antibody and it's not a signaling antibody. So this has to be determined first. Otherwise, your interpretation is going to be totally different. So that is why I actually talked about this slide here. All right, so there's some questions here. We are purifying FC with protein A elution. So does it mean the binding is associated with FC? Yes. Okay, the answer is yes. Other question is, does 3 CDR, well, do 3 CDRs mean the antibody will bind to 3 different antigens? No. 3 CDRs from light chain and 3 CDRs from heavy chain will constitute one paratope. And this one paratope will have specificity towards one epitope. Okay? 3 combined. Rather, 6 rather. This is because of the combinatorial diversity that you get from having a heavy and light chain CDRs. Okay? 6 CDRs make paratope that will bind to an epitope. Okay, so they got for negative selection. Well, they actually did not get it for negative selection. Yes, this was actually, see, negative selection and positive selections are kind of known. They were known for quite some time. Peter Medawar and many other scientists, actually they showed it long back. Okay, so what they got this Nobel Prize for was to use this immune checkpoint inhibitors like PD-1, TIM3, and basically how you can tweak around signaling through these receptors to reinvigorate the function of your immune cells. So that's the area which was recognized for Nobel. What are the 60 amino acids and 110 amino acids? Well, 160 amino acids actually are in the loop. Okay, and then you have the disulfide bond, and then some sequence is here and some sequence is here. So this whole thing actually is considered immunoglobulin domain. Can blocking antibody be used for stopping immune response via TCR? Well, yeah. So for example, if you are targeting some activating receptor, yes, they can be. Well, rather, to tell you the truth, actually, we have some antibodies we generated in our lab. They actually have phenomenal functions, like either promoting immune responses or inhibiting immune responses, either at lymph node or at tissue sites. And we actually have generated them in the lab using this technology that we call as phase display approach. Anyways, so any more questions?
Sir?
Yeah.
So I have 2 questions from the past lectures. So one is regarding the epitopes. As you told that, I mean, whatever we discussed till now is regarding the epitopes of proteins, like, you know, the epitopes in proteination is in nature. So you told that also epitopes can also be other biomolecules like carbohydrates and all. So in that case, I mean, would they exist, you know, in one place? And secondly, how would the interaction with those epitopes and the paratopes of antibody achieve? So
yeah, well, see, even in carbohydrates you have these side groups, right? So look at the structure of these carbohydrates. You have these exposed regions, right? So, and if those things come together and make one epitope, so they can be recognized by your antibodies. But as I said, the most epitopes, rather almost 95% of the epitope for antibodies or for TCRs are coming from proteins, right? But this is not to say that lipidated antigens okay, protein molecule which is lipidated or carbohydrate or glycosylated or the carbohydrate regions of an antigen, they can also contribute heavily in antigen antibody interaction.
And so secondly, it's regarding the immunoglobulin fold. So is the fold present? So immunoglobulin is a basically superfamily. So is the fold present in other entities of the family also, or is it specifically?
Yeah, exactly. So that is why the other proteins are also called as the member of immunoglobulin superfamily. So I'll talk about that. So don't worry. Okay. So I'll have one slide or maybe two slides dedicated to that what are different molecules which actually are part of the immunoglobulin superfamily, and they are not antibody molecules. Rather, TCR is also part of immunoglobulin superfamily.
Fine, sir. Thank you.
Yeah. If the blocking antibody interacts with inhibitory receptor on NK cell, can they lead to a mass apoptosis? Well, why do you think so? Well, if it interacts with the inhibitory receptor on NK cells, probably NK cells will become activated. Yeah, after activation, so obviously these cells are going to die. Maybe you're partially correct, but the major function is that this is going to enhance functionality of NK cells. All right, so I think with this, I think we can stop. So then I will talk about basically the effector function of antibody molecule and then antigen antibody interaction and so on and so forth, right? So up till this point is going to be your semester, rather your course for your first mid-sem exam, up to slide 137. And I'm going to upload these slides by the end of the day today. Your exam, I think, is on Monday, right?
Yes, sir.
Yeah. So at 9. So I will schedule it and send you the information. Through what? Can I do it through WhatsApp message or on Moodle page? Maybe I'll put it on Moodle page, right?
Fine, sir. Yeah.
Yeah. All right, fine. So let's stop it here.
So will we have class on Thursday, just to confirm that?
Well, that actually depends on you. I'm okay with Thursday's class because by Thursdays, rather by Wednesday, your exams will be over, right?
Yes, sir.
So let me know if you want to have this class on Thursday. My preference is yes, let's have it because we actually have to cover quite a lot.
Sure, sir.
Yeah. Yeah, exam will be in the Google form. Yes.